All right, I think we're going to go ahead and get started today. So it's my pleasure to introduce Morkos Henen, who's going to give a talk about, I love his title, I was just talking to him about that, about chasing cargo over microtubule tracks. Just to give you, this is so loud. Just to give you a little background for Morkos, because he's a relatively new addition to our faculty here at the university. He did his PhD at the Max Perutz Labs in Vienna in Austria, and then followed that up with a fellowship at Marie Curie in Lille, France. And in all of these positions, he's been using structural biology approaches, primarily NMR approaches, to do sort of drug development and drug design. He then moved to the States, where he worked as a postdoctoral fellow, first at the Health Sciences Center in San Antonio, but his PI moved to Pittsburgh, so he then finished up his postdoctoral work in Pittsburgh, and again, mostly around looking at protein interactions by NMR and other structural methods in order to design or think about new pharmacologic interventions. And he worked on some of the key sort of players that we know in cancer, like TGF-beta signaling and Wnt signaling, so the interactions between beta-catenin and TCF-LEF. And after he finished his postdoc, he moved to the University of Colorado, where he has been working in the, as a research instructor in the Vogeli lab, and it's there that he's looking at dynein-mediated cargo and transport, again, using structural approaches. So with that, I will turn it over to Morkos.
Just have to switch the mics. Thanks for the introduction, and thanks for inviting me. So I was thinking today about a completely different unprofessional title. I was going to say it's a 3 countries journey, because I will show data from United States, from Vienna, and from France. So I will start with the United States, and then I will take you back a bit in the time and space to Vienna, and then somehow afterwards to France as well. So since I joined the Vogeli lab about 2 years here in Colorado, I have been working on the dynein system. And as you can see here, this alien mainly on a small part of dynein, because dynein itself is pretty huge. So only a very small part of it I have characterized. I will show in vitro, in vivo data, which we just published in PLOS Biology a few weeks ago. In the second part of my talk, I will still be on the dynein system, but maybe shifting a bit to part of the dynein complex, which is called spindly. It's kind of emerging new protein as a target for cancer now. And we are also working on characterizing this using NMR and using in vivo CRISPR-Cas assay in C. elegans. The last part of my talk, and this is the part where I will take you back in time and space, this is the drug design approach, where I will talk about a couple of projects I worked on in Vienna and in France, where we have established like a very, very nice sensitive technique for drug discovery, which is being adopted a lot now in pharmaceutical companies. I'll show you how we can use that, very nice for targeting cancer. So starting with the first part, which will be about dynein. So what's dynein? I'm sure most of you are aware about dynein. So dynein is a motor protein which is moving over the microtubule, which is so this one. Yeah, exactly. So this is dynein. It moves over the microtubule to the minus end of the microtubule, while the counterpart or the enemy of my protein is called kinesin, and kinesin carries the cargo over the microtubule, moving in the complete opposite direction, which is the plus end of microtubule. It has been for a while believed that there is kind of tug of the war between the two, because both proteins, as you can see, they move in opposite direction. So for bidirectional cargo, it was believed that one of them has to compete or to win the other. I think this theory is kind of not so solid now. There is even now theories that they kind of cooperate, and then at the end, just it's a regulatory mechanism which makes one of them goes in one direction and not the other one. But I mean, I will not talk much about that. I will focus on dynein. This is the cover of Science magazine in 2011, when they found the structure of the ATPase domain. This is the head, this is the motor of the protein where all the energy produced to give the protein the power stroke in order to move. And then the protein also has these legs and the feet moving over the microtubule. What they didn't show here is the tail of the protein, and that's where I come, hopefully. So this is the structure of dynein. Dynein is pretty huge, complex. It's 1.4 megadalton homodimer. This is, again, the heads. That's where ATP is binding to give the power stroke. These are called the stalks. These are the legs of the protein. These are the feet, which are microtubule binding domain that bind to the microtubule to start moving. And then this is the tail part, and the tail part has this intermediate chain, this guy here, and then the lighted chain. And then the one highlighted in yellow is called the light intermediate chain. Just to tell you here, light intermediate chain used to be like the least characterized part of dynein. So usually it was kind of overlooked, but that's what I'm going to talk about mainly today. This is more for fun. I mean, don't take this so serious, but it's published in Nature still. So I mentioned that dynein is moving toward the minus end of microtubule, right? So this new paper here, they wanted to do something like, I would say it's funny. They just wanted to see if dynein is possible to move to the positive end of microtubule instead of the minus end of microtubule. And for that, they engineered a specific type of dynein where they increased the length of this stalk, this yellow part here. They added 7 residues extra, and then they mutated 2 conserved proline in the microtubule binding, these 2 dots here. And they found out actually that this changed the directionality of moving of dynein. So now dynein, according to this EM and also some simulation they did, is moving to the positive end of microtubule. But in reality, actually, this doesn't happen because the length of this stalk and these 2 proline are conserved among all species. So this is kind of something just to show that dynein is exclusively moving toward the minus end. So what's the problem with dynein? I mean, nothing in life is easy, as well as dynein as well. So dynein doesn't move processive over the microtubule on its own. Dynein has a problem. It cannot move on its own over the microtubule. It needs cofactor called dynactine. Why is that? So dynein itself, this is the tail of dynein on top here, and these are the legs. So you see the legs are like crabbed like that, which is called auto-inhibited state of dynein. So dynein cannot move this way. So in order to get out of this auto-inhibited state, it has to stay in this parallel leg situation where it can move processive over the microtubule. And in order to stay like that, it has to bind a cofactor called dynactine. This is the one shown in brown here. But again, there is additional problem. It needs dynactine in order to get out of the auto-inhibited and to be able to move over the microtubule, but binding itself between dynein and dynactine is kind of transient. The affinity is pretty weak to keep this complex together. So here we need a glue, we need a protein in between these two to connect dynactine to dynein in order to form this ternary complex. And this glue in this case is called adapter, and I will be talking a lot also about adapters today. So adapter is a protein which connects dynactine and dynein to form this stable ternary complex. And these adapters, beside actually forming the glue here, are also the cargo carrying protein. So part of this adapter, the C-terminal usually of this adapter, is the part which carries the cargo while moving over the microtubule. There are a bunch of adapters known, and these adapters are usually cargo specific, as I'm going to show now. So just showing here, this is EM structure of the complex. So if you look at this cartoon here, you see we have only one dynein, which means we have only like 2 legs, 2 feet, and they bind to one adapter, one dynactine. But in a recent paper, it's also, I think, in Nature. Yeah, it's in Nature also. So here they found out that in sometimes it's not only one dynein which forms this complex, but we have here 2 dyneins. So you see we have 1, 2. This is one dynein dimer, and then we have these ones in blue. This is a second dimer. And they found out that some adapters, which are this protein in yellow here in between dynein and dynactine, are able to recruit 2 dyneins instead of 1. The question was why some adapters are doing that and some adapters are not doing that. And the theory until now, it seems that some adapters actually need to have better force and more speed in order to counteract the effect of kinesin. A kinesin, as I mentioned, is the counter protein which moves to the positive end of microtubule. So in order to increase the force of this movement, some adapters need to have two dyneins. But this is not the case for all complexes and not all adapters, only specific adapter. But this is kind of new still, so it's not clear that this is the exact reason, but this is one explanation, that some need really to have stronger force, more speed in order to counteract kinesin movement. Okay, there is one part on this structure here they were not able to show, and this is the part I'm interested about. So if you look at the EM structure here, this is the adapter, and this is one of the dynein parts, the tail of dynein. And in the EM structure, they show that there is a contact between the adapter and dynein, and they were not able to see this by EM. And this part, they believed in this paper, it's LIC. LIC is the part on the tail of dynein, light intermediate chain. So from now on, I'm talking also again a lot about LIC. So what is the problem with LIC? Why they were not able to see LIC in the EM structure? Before I talk about that, LIC is also kind of emerging target for drug discovery now, targeting cancer. So there is this new compound which was just described last year. It's called denaristine. This compound was found actually to inhibit the hedgehog cycle. So the hedgehog cycle is overactivated in cancer, in some specific cancer like basal cell carcinoma, for example. And it's kind of target for drug discovery against cancer. And there are already drugs in the market which target this SMO receptor in the hedgehog. But there are other events which happened downstream to the SMO where dynein is playing a very crucial role for the transportation. And targeting dynein here would be an interesting target for drug discovery against this kind of cancer. When they came with this compound, they said it binds to dynein, it inhibits the hedgehog, but they were not sure which part of dynein it binds, but they have a theory that it might bind to LIC. LIC is this part of dynein tail that I'm focusing on. Second, very new paper as well, in colorectal cancer. They found out that there is overexpression of LIC particularly. As you go with the stages of cancer in colorectal, it seems that LIC expression is getting more and more. Besides also that LIC here is playing a role in the cell migration, which means it can play a big role in metastasis in cancer. So it's also kind of considered, according to this paper, that it's also target for cancer discovery or for cancer treatment against colorectal cancer to target LIC. But again, we still have the problem. What is the problem with LIC? Why it didn't show up in the structure before? So LIC has what's called a disordered C-terminal. So you probably heard about the disordered protein before. So disordered protein are proteins which are not able to adopt any fixed 3D structure. They are always like tumbling in the solution like that. They don't have a fixed structure. And these kind of proteins are kind of problem for EM or X-ray structure because you cannot really get structure. There is no structure, basically. But what's important is that this disordered part of LIC, the C-terminal part, this part here, which has never been characterized before, is actually the part which binds to the adapters. So these are a bunch of adapters here, and it seems that they are all binding to this disordered part of LIC, but there was nothing to describe this interaction on atomic level because the C-terminal of LIC is disordered. So here comes NMR. So what's NMR? NMR is nuclear magnetic resonance. It's probably this weird term. When I say NMR anywhere, people just get scared. Don't be scared. I'm not going so deep into NMR today, but I'll touch base. Not really. So why NMR for disordered protein? NMR is the only method that you can characterize disordered protein on atomic level. You cannot do that by X-ray. I mean, with all the respect to these other techniques, they are great. But just if you have a disordered protein and you want to characterize this, we are your guy. You have to come to us. So, but still, I would say NMR for IDB, although it's kind of well established now and there are many groups working on that, it's still emerging new field of discovery, because I was telling our facility manager like a couple of weeks ago, when I was doing my PhD in Vienna, also working with disordered protein, we were always calling them in the lab unstructured protein. And everybody in the lab was just saying that. And then one day, I don't know how this happened, we were in the morning, nobody to say unstructured, it's called disorder. I don't know how this transition happened, but then I was reading this paper, it's called What's in the Name. It's adopting the Shakespeare play, Romeo and Juliet. Sorry, I'm going. And they are also confused about naming these proteins. So even they call them dancing protein. So these proteins, disorder proteins, have been going through so many nomenclature, but I think the name, which kind of, I hope stays a bit now, is disordered protein or disordered regions. Again, these proteins, they have high degree of flexibility. They are playing an important role in diseases. For example, people of you who are working on alpha-synuclein or tau, they are involved in neurodegenerative diseases like Alzheimer. Tau is considered a big target to understand Alzheimer's disease now. And again, these proteins, although no structure, really 3D structure, they can still be assigned and understood on atomic level using NMR. This is the machine on RC1 South basement. I don't know if some of you saw it, probably. It's like a spaceship. So this is a 900 machine. But believe me, I mean, it's big like that because we fill it with liquid nitrogen and helium, but the probe itself is really small. And we put only 300 to 500 microliter sample in all of that. Okay. Just a quick thing about NMR. So I mentioned that the C-terminal of Lick, the one that has never been characterized before, is disorder protein. And this is one example of what we can see by NMR. So here we have a 2D NMR spectrum, but just for simplicity, every peak here represents one single amino acid in this protein. And the position of every peak, for example, this alanine is here, while this alanine is here because of the difference in the chemical environment. So every residue experiences different chemical environment, different secondary structure, different dynamic is showing on a different place on this whole scale. And this thing as a whole is called a fingerprint 2D of the protein. And this is what we can use actually to study interaction and structure. So using also NMR, I mean, these are relaxation experiment NMR, which don't worry, I'm not going to talk about that at all. But just from these data, we were able to show that this Lick C-terminal, which is a disordered protein, is not really so disordered, which is kind of common also for disordered protein. What does it mean it's not really disordered? Looking at our NMR data, when it's translated into a model, this is the model that came to the protein. So the whole protein looks like a disordered protein, but there are two areas in the protein which are adopting helical propensity. They have helical, again, it's just propensity. It's not a fully formed helix like what you see in normal folded structure, but it has a propensity here for two regions in the protein. So the whole protein seems to be super flexible except these two parts. The question that we came after, what is the role of these two helices? What these two helices are doing here? And for that, we looked again at our fingerprint. So I remind you again, this 2D spectrum is a fingerprint of the protein, which means if I add any binding partner, for example, to this protein, right? And if it binds, it will change the chemical environment. And this change in the chemical environment will be reflected in a change in these peaks. Either they get less intense or they move from one place to another. So we can actually map the binding site using these NMR experiments. And that's exactly what we did. So we know that Lick C-terminal is binding to the adapters, right? So we titrated many adapters. These are 3 different adapters with the protein. And we were able, if you look at this histogram, we were able to map the exact binding site here. So we have 3 binding sites on Lick that are binding to the adapters. The first site is what we call it helix 1. This is the first helix. you remember I told you we have 2 helices predicted on this protein. So the first helix is the one which is showing the most and the biggest effect upon binding. You see this by the peak intensity reduction. And then we have a second region, which is just a disordered part N-terminal to the first helix. So this is helix 1. And then N-terminal to this helix, we have also additional part which seems to be important for binding. And then the third part is helix 2. And this helix 2 is also involved in binding. So we show it for so far now by NMR. I'm done with NMR here, by the way. That the protein is disordered. It has 2 regions which are having alpha helical propensity. Upon binding to adapter, there are actually 3 regions important for binding. And these regions are helix 1, helix 2, and also this particular region N-terminal. The interesting part is that this disordered N-terminal part is conserved among all species, all vertebrate particularly, sorry. It's conserved in vertebrates. Okay, we use the SPR and the MST to see the effect on the KD. You know, here you can, by SPR, MST, you can measure the kinetics of the interaction. And the results here, I have to say, I mean, I can say it and maybe you will be convinced, but it's not convincing for me. So what we did is that we have the full length lick and we get a KD of about 5 micromolar. And then when we remove helix 2, the KD seems to be the same. When we remove helix 1, which we believe is more crucial for binding than helix 2, the affinity gets weaker by 4 times. This is not so significant, I know. Okay, so how did we address that then? So here we went to the pull-down assay. And in this pull-down assay here, we have several constructs where we were having like the first, if you look at the first construct, this is the whole protein. Then we removed the helix 2, and then we removed the helix 1, and then we have here mutations in helix 1. And what we can see by pull-down assay is that when helix 1 is there, and this N-terminal part is there, the binding is okay. Once you remove helix 1 or mutate helix 1, the binding is just gone. So this shows that helix 1 is indispensable for binding. Then comes the second part here. So we wanted now to see the effect of this N-terminal part. This is also pretty exciting. So the N-terminal part, as you go shorter, you see the affinity gets less and less, which means that this N-terminal part is really, as NMR showed, is playing an important role for binding. And when you get rid of this N-terminal completely, the affinity of the binding is just gone. What about helix 2? When you have helix 2, helix 1, and no N-terminal part, there is still modest binding. So if you allow me to rank these 3 regions in importance for binding now, and this is what we will show in C. elegans in a moment, we have 3 regions for binding, helix 1, helix 2, and this N-terminal part. Helix 1 is indispensable. Helix 1 is gone, the affinity is gone. It doesn't bind. The second in ranking would be the N-terminal part. As this gets shorter, or if you get completely rid of it, the affinity is also, the binding is also gone. Then comes the helix 2, which seems to be kind of dispensable, not so necessary for binding. And that's also, again, what our in vivo data show. So based on these data now, we have 3 binding sites. We know this. We wanted to test this in vivo. We wanted to see this in animals and C. elegans, what happens. So we switched to our collaborator, Reto Guzman. He's working in Porto, Portugal. And he took my NMR results and just applied these kind of mutations using CRISPR-Cas9 in C. elegans. And I came to him with the question, my ranking, is it correct or not? Is helix 1 really important, more important than helix 2? What about the N-terminal region? Just unfortunately, the N-terminal part, which seems to be the flexible part, is not conserved in C. elegans. So we didn't test this part in C. elegans, but we tested helix 1 versus helix 2. So animals which are lacking helix 1 or have mutations in helix 1, they were able to develop to adulthood, but they were sterile, so no progeny. And also the motility here was kind of hindered, so this is a motility assay, so they show less motility. Also on top of that, they have developmental problem in the gonads, in the tails. More importantly, they have shorter lifespans. So animals which don't have helix 1 are having between 25 and 35% less lifespan than animals which have helix 1. Okay, we know that this protein is part of the dynein machinery in general, so it's involved in cargo transportation. So now we see that the animals are suffering, it seems. We wanted to test more in depth about the cargo transportation. Does the cargo transportation get also affected by mutation in this helix or not? So in this case here, we just have the animals. So the microtubule, in this case, this is the positive end of the microtubule. It's the axonal tip of the neuron. And then this is the negative end or the minus end of the microtubule, which is the cell body of the nerves. So normally dynein would move to this direction, would move the cargo in this direction. So using GFB to label the neurons themselves and MK to label the early endosome. So early endosome is a very important cargo to be transported by dynein. And what we are seeing here is that we see the transportation of early endosomes in animals which are lacking this particular helix. And as you can see here in the normal animals which don't have any mutation, the transportation seems to be going smooth. These are the neurons. But once you have the mutation in helix 1, you see this it reminds me always of traffic jam in Denver. So it looks like we have a big chunks of early endosomes accumulating in the neuron because the transportation is just hindered. Because again, this part of dynein is what binds to the adapters. So if you actually hinder the binding to the adapters, the protein cannot move processively normally anymore because of this particular mutation. And then this chymograph here is also showing here. So if you look at the first one, you see like everything is just moving smooth and liquid. But once you have mutation in the first alpha helix, you start to get these chunks of the early endosomes accumulating. The transportation is just affected. I hope this video will work. So here again, in this video, which is just a translation of the chymographs, we can see there is normal transportation going on in animals which are having helix 1, helix 2. And once you get this mutation, you see here the transportation is kind of slowing down. There are this accumulation of the early endosomes. So this again shows that helix 1 mutation did the following. The animals, although they were able to develop to adulthood, they die early. They don't have progeny. They are sterile. And the transportation itself is also affected pretty much. Okay. And that agrees with our NMR data, which shows that helix 1 is indispensable. So what about helix 2? So when we mutated helix 2 here, again, from NMR data, we know that helix 2 doesn't seem to be super crucial for binding like helix 1. And that's exactly what's happening here. So animals lacking helix 2, they don't have any transportation problem. Everything is moving smooth. They develop to adulthood. They have embryos. They are fertile. So everything seems to be fine. Only one problem. When looking at the mitotic division, the very first mitotic division of the embryo here. So in animals which don't have this mutation, how did I go that far back? So animals without this mutation, you see here, this is a spindle at the mitotic division. It has to get parallel to the anterior-posterior axis of the cell line just at the nuclear envelope breakdown before the anaphase in order to get the separation of the two chromatids. Upon mutation of helix 2, you see here there is a delay in this alignment of the spindle. So if you look at the angles, which in ideal scenario, it should be 0 degrees at the nuclear envelope breakdown. So this thing should be just parallel to the cell line. But this doesn't happen here. We have a delay in this positioning of the spindle. The angle recorded was around 45 degrees. But what's interesting is that this, although of this delay, it's still the spindle was getting to its normal position, and then the separation was happening normally. So if you look at the video on the top here, this is a normal division here without any mutation in helix 2. So the spindle gets normally aligned or parallel to the cell line, to the axis of the cell. And then upon mutation, we just see a delay in this spindle. So it takes a while longer than the first case to get parallel with the anterior-posterior cell line. So this shows that there is effect on the mitotic division here because of the mutation. And in a second, I will show what dynein is doing for mitotic division. But then it's not so crucial. The animals are fine. The animals are alive. They don't die. They are fertile. So in a quick summary for this part here, we know that dynein is moving over the microtubule, going to the minus end of microtubules. We have 3 regions on LIC, which has never been described or characterized on this level before because it's a disorder protein, so they needed NMR. They needed me, apparently. And we have 3 regions here, helix 1, helix 2, and this N-terminal part. We showed that helix 1 is indispensable for binding. Helix 2 seems to be less dispensable. I mean, we have a lot more results than that. We just published that in PLOS Biology, so if you want to read more about that. Okay, in the second part, I talked that dynein, dynactine, they need adapters. So there are a bunch of adapters, and these adapters are cargo-specific, which means every adapter of these is transporting different kind of cargo. So for example, big D is mostly specified in transporting RNA. Spindly, which is the one I'm going to focus on now, is actually playing a crucial role in the mitotic division, especially at the anaphase. So spindly, the part which is shown here in a box with the lines, it's the part which binds to the dynein, dynactine. It's the part which is here. But this C-terminal part is the part which binds to the cargo. And the cargo in this case is part of the kinetochore during the mitotic division. So let's look at that. So if you look at the kinetochore, you have these 2 chromatids. Before separation of these 2 chromatids, before the cell entering the anaphase, there is something called sac. It's a spindle assembly checkpoint. This sac is super demanding. It has to be satisfied. How to satisfy the sac? The sac gets satisfied. Okay. The sac has to be satisfied by recruitment of microtubules. So if you look at the outermost layer of the kinetochore, before the anaphase, it has to recruit microtubules. And when this complex is formed correctly, what's called the KMN, the kinetochore microtubule network, then the cell is now ready to enter the anaphase. This whole thing, the outermost layer and the sac, has to be removed in order to allow the cell to enter the anaphase. And this removal is happening mediated by a complex called RZZ, and this RZZ is what binds to spindly. So this is spindly, this is dynactine, dynein, so this is the part I've been talking about at the beginning. And then the C-terminal of spindly binds to RZZ, which is called rod ZW, it's Welch, and this RZZ complex binds to the sac. So what happens is that this whole machinery here just takes away, strips away RZZ and the sac over the microtubule away from the kinetochore, and this silences the sac, and when this happens, the cell is now allowed to enter the anaphase. So again, looking at the spindly C-terminal, which is the current project I'm working on, and we are also getting students through the Cancer Center who will work with me on this project. So spindly C-terminal is another IDP, another disorder protein. This is again a fingerprint of the protein. In this case here, using NMR, we were able to get this model which shows that the protein is mostly adopting 1 helix in the middle. I mean, it was also predicted to have 2 helices, but actually my NMR data doesn't agree on that. We have only 1 helix. And what we are working on now is that we want to understand how the interaction between this spindly C-terminal and RZZ is happening. So this is kind of work in progress. I'm not going to show you much on that. So for example, this is the mapping of the binding site between spindly and that's Welch. And we are able to see these residues involved in binding. Our collaborator now is doing mutation on these residues in C. elegans to see the effect on the mitotic division in animals. This is SPR data. I'm still working on also characterizing the interaction with rod and with sac as well, because nobody actually showed before that sac is interacting with the spindly. So that's what we try to see. Okay, now my favorite part. Will not be long, I hope. Okay, so drug design approaches. So we have now two regions or two places in the systems I have just talked about which are considered target for drug discovery. So I mentioned that lick here is overexpressed in tumors. It seems to be a kind of emerging new target to understand cancer or to target cancer. Also, the C-terminal of spindly, which is binding to RZZ, it seems to be overexpressed in tumors, especially in lung cancer. So these both are kind of targets now to design drugs. We didn't start on that yet, but I will show you very exciting old results on drug design using NMR. So why NMR in drug design? You know, most of companies now, when they start any drug design approach, they have a bunch of compounds. They usually start with library of like 12,000 compounds, small fragments, and they have a target. And they want to measure interaction between these compounds and the particular target they have. The first problem they usually face is the weak affinity between these fragments and the protein. Usually this is the very first problem people see when they have a drug discovery, that the affinity is weak. Sometimes it's even in the millimolar affinity. So other methods might actually miss on that. They cannot probably see that. But NMR is super sensitive for drug discovery, that even weak affinity ligands can be seen binding to our target using NMR. And then we can also go with optimizing these fragments. So we have millimolar affinity. So you can actually go with growing or merging, or you can have two fragments, merge them to each other, or using chemical synthetic chemistry, you can grow one fragment, and then you can go to NMR again, measure the affinity again, hopefully getting better affinity. So this technique is kind of using NMR for drug discovery is kind of standard now for starting drug discovery projects in company such as, for example, AstraZeneca. So what can we do by NMR? We have two ways to do NMR for drug discovery. The easy, simple way, it's called the ligand-based NMR. Probably people who hear about NMR, they know that we have size limitation on NMR. Usually large proteins are a problem for NMR. We cannot see them. But this is not the case for ligand-based. Give me whatever large protein you have, it's still fine to do ligand-based NMR, because ligand-based actually might be even advantage for larger protein. Also, we don't have any problem with the concentration. You can start with as low as 10 micromolar protein in the NMR tube. Ligand, maybe we need more. We need 1 millimolar, 2 millimolar, maybe a bit more also if you have weak affinity really. But again, and then the protein doesn't have to be labeled. Probably also people who hear about NMR, they know that in NMR, we label the protein 15N or C13. You don't have to do that for ligand-based NMR. Pretty simple, 300 microliter sample. That's what I always say to our collaborator. 300 microliter sample, 10 micromolar protein, unlabeled, just LB produced or whatever if you use mammalian cells. And then for that, we add excess of ligand, which can be about 1 millimolar. And then we can go with a bunch of experiments. And what we can get from these ligand-based NMR experiments here, we can see, does it bind or not? This is a simple question we can answer. The second question we can answer also is the KD. Does it bind with a strong or weak or medium affinity? The important question which we can answer is pharmacophore mapping. We can detect which part of the ligand is actually crucial for binding. And this is actually a very important first step for drug discovery. So you don't need to hassle so much with so many fragments to find out which one you need to design around. We can just tell you this is the pharmacophore, so stick to this part and go with that. The second method, this is more complicated. It's a target-based NMR drug discovery. In this case, now we have the limitation of the size. The protein has to be in the size limit, which is around 50 kD. And also the protein has to be labeled, 15N and C13 labeled, so this is a bit costly, of course. But the advantage in this case is that you can map the binding site. You can see which amino acids exactly the ligand bind to. So quickly, I will not talk much details about this project. So this is part of my PhD project in Max Perut's laboratory in Vienna. And in this project, we were working mainly on studying beta-catenin, which is the main key player in the WNT cycle, and it's considered an important target to understand or to target colorectal cancer. It's overexpressed in colorectal cancer. So beta-catenin has so many ways to escape degradation going to the nucleus. It binds to this transcription factor, which is called TCF4. And when it binds this leads to the transcriptional activation of this cycle, which is kind of overactive in cancers. So what we wanted to do, we wanted to find ligands that disrupt the binding between beta-catenin, the one shown in green here, and the transcription factor is the one shown in red here. And for that, the first step we did, we started with a small compound which was only predicted based on algorithm, which was also developed by my former PhD supervisor back in Vienna. And when we did our NMR experiments, which I'm not going to talk much about, we were able to show that this predicted fragment, okay, it's binding. And then we were able also to get the KD, as I will show you in a second. This was pretty weak affinity. It was in the millimolar affinity. But what's important is that we started to play around a bit this ligand by testing similar ligands to this guy. And what we were able to see is that we were able to detect the pharmacophore here. So this particular part of the ligands, I tested here like tens of compounds. I'm not showing you everything. But all of these compounds I tested, they shared this pharmacophore. So we showed that this phenoxybenzene moiety is essential for binding to beta-catenin. What also we did by NMR is that we were able to go from millimolar affinity, which is probably something would have been missed by any other techniques. Again, this is not a drug. It's a very preliminary compound to understand what drugs can be for this protein. And then we were able, using growing for we were able to go to ligands which are 100 micromolar. Again, it's still not super strong, but we improved the affinity. And what was more interesting is to find the pharmacophore. Again, in these experiments, I had only 10 micromolar protein and 1 millimolar ligand. This is too small amount of the protein. You don't have any trouble with that. Okay, now we came with the question, how do we know that these ligands disrupt the interaction between beta-catenin and TCF4? So I mentioned that if we want to map the binding site, we need to label the protein, and we need to go with hardcore NMR method. The problem with beta-catenin is 85 kilodalton protein. This is too big for us. So the problem here is that we could not do regular or standard NMR, but we did a trick which X-ray people, probably if there are X-ray people here, they know this technique. It's called C13 reductive methylation. So when X-ray people face the problem that the protein doesn't crystallize, they try to reduce the movement or motility. And for that, they increase the hydrophobicity of the lysine side chain. So this is just a chemical reaction targeting lysine. In this case, we interact with 13C formaldehyde in presence of reducing agent. And so we end up with a protein where lysine is only labeled C13. And this way, we can measure NMR for that. We cannot measure regular 15N NMR and see every single residue. This is not possible for this kind of size of protein for NMR. But what we are able to do here is just to measure lysine amino acid. And of course, as you can expect, for looking at the structure, most of lysines are just on the surface of the protein, so they all appear as this big blob here in the middle, while lysines, which are in the middle or in the core of the protein, they are the ones which appear distinct here. So this is beta-catenin alone. This is beta-catenin with TCF4. We see only one single lysine shifting or moving. So we know that this lysine is in the binding site between catenin and TCF4. And then now we tested the ligands that we came to by our NMR method, and we see that the same lysine is shifting also upon binding to beta-catenin, which is telling us that these ligands and TCF4 are sharing similar site and considered a potential inhibitor to disrupt the binding between beta-catenin and TCF4. Again, we were not able here to map the binding site really completely because of the size of the protein, but at least we were able to get a piece of the story that our ligands are not so far from disrupting this complex. Okay, I think I will skip this story. So this is a French story. So the French guy is not here today, so that's good. I can skip that. Okay. Although this is pretty exciting one, but I have to skip it. So in a quick summary here, what I showed you today, my problem is that whenever I go to any talk, I say NMR and just people leave. I'm happy that you didn't do that today. So NMR is probably like complicated technique to understand on the quantum mechanic fundamentals and things like that. But it's not necessary that everybody comes to NMR has to understand all these complicated fundamentals, because simply that, if you have a target and you want to test a specific ligand, just give me this target with some small amount of the ligand, and by NMR we are able to do 2 hours, 3 hours experiment and tell you immediately if it binds or not, and I can tell you if there is particular part of the ligand deep inside the pocket or part of the ligand outside toward the solvent more. This is simple as I say, really. You want to go deep into NMR, you want to map the binding site, then we will start talking about making labeled protein and then getting fingerprint and detect exactly which residues are involved in binding. Just to tell you also how powerful is that, we are working now with 2 groups on the campus here. The group of Richard Johnson in the renal division, and also the group of Sean Colgan in the gastroenterology. And in both cases here, they have targets. Unfortunately, I have results, but I cannot show you because these targets are not for cancer. So since we are in cancer event now, they are more for inflammation. And we were able also to show exact pharmacophore targeting a protein like myeloperoxidase with a group of Sean Colgan. And myeloperoxidase is considered a big target for anti-inflammatory drugs. Also, collaboration with the group of Richard Johnson, we are targeting AMBD enzyme. This is more for diabetes. We are also working on KHK. It's a target also for diabetes, metabolic diseases, and for alcoholism. So for these 2, we are still taking on the project. They gave me the protein, they gave me the ligand, and they get the answer next day. It's simple as that. Also, in collaboration-wise, we are collaborating with the group of Kirk Hansen and the group of Ray. With Ray, actually, we are also starting anti-cancer drug discovery project targeting 6-1. I was talking now about that. But this is still taking off. Hopefully, Genevieve will give me protein soon. So I probably, for acknowledgement, I started a bit on the... Also, one thing I didn't mention here, since we don't have expertise in the lab to do cell line assay, you probably see we are hardcore NMR spectroscopists. We do a lot of in vitro assay. So the collaboration with these guys is pretty nice about that because they are really, really experts in using cell lines. So I think they will take my compounds, which I show by NMR, they bind or they are interesting for bind, and they will take this to the next step by assaying in cell line to see the effect of these compounds there. Also, collaboration with Reto Guzman in Portugal. He's the expert on the C. elegans. So again, I don't have expertise in C. elegans, and he's the one who's taking my NMR results and just translating this in animal models. I kept this to the end. So this great group, actually, my mentor, Beat Vogeli. By the way, Beat is Swiss. So his family name, Vogeli, here means a small bird in German. So the logo of the lab is this nice bird here. In the group also, we have Alexandra. She's working on PIN1, which is a PPIase protein. It's also important for cancer, and she will be giving a similar talk soon. She's really having so nice results, and she's presenting that in one of the biggest NMR conference today in California. Parker, who is our recent graduate student in the group, he's working on RNA. He's the RNA guy in the lab, highly experienced guy. And he's working on this zRNA. It's a very interesting, exciting story. And I think also they are presenting this story in the NIH end of this week, right? Max and Lauren, two former rotation students in the lab, both of them worked with me on this dynein project. And actually, Max shared a paper with me, the BLOS biology on this dynein system. And Lauren soon, hopefully, also sharing something with us. NMR facility, David Jones, he's a great, great NMR scientist. I mean, I cannot thank him enough, really. Biophysics course, Sean as well, also cannot thank him really enough. Unfortunately, Sean is taking care of the biophysics core, but he's also leaving us soon. I don't know how will be the core after him. I mean, okay, I will not cry. It's fine. I also want to thank Ilan. Ilan is an NMR spectroscopist in our group, and discussions with him are always really fruitful. Quentin, he is working with Parker on the RNA project, and also Quentin is having kind of drug discovery project in mind targeting RNA, but okay, this is still speculations. We don't know yet. I want also to thank really the Cancer Center. James De Gregory, I used to go to his office every time asking him, how do we target cancer with that? What do you do with that in cell lines? Because these are the things that I really don't get. Dr. Haida, she's very supportive. I mean, she nominated me actually for Sam Baldrick cancer grant, which I submitted, hopefully you get it. Jill, I mean, I cannot thank Jill really enough. She is super supportive. And thank you all for attention. Thank you. Sure. Yeah, yeah. Yeah. Yeah.
Are the interactions the same for each depth and line curve? Is there any regions that might be more combining one reactive sample compared to another one?
I like this question. Okay, so there is part of the project which I didn't show much. So we tested 3 adapters from our side by NMR. And for the 3 adapters we tested, it seems that for all 3, the binding is going in the same way, the 3 regions, right? But our collaborator, he tested an adapter which is called RELB. It's in the transportation of the vesicles. And he showed that for this particular adapter, the story seems to be different. It's only the helix 1. Helix 2 and this disordered N-terminal part seem to be doing nothing. So helix 1 is only sufficient for binding an adapter like that. So RELB is actually one of the, I should have mentioned that. It's one of the projects we are running in the lab now. So we want to understand why RELB is different than the other adapters in binding lake. But there is a problem, fundamental NMR problem here. RELB is a coil. And these coil are usually, it's like a cylinder. So they relax pretty fast as the signal goes pretty fast in NMR. So we are still optimizing on that. But you're right. I mean, it doesn't seem, for the 3 I tested, it seems the same, but it's not the same for every adapter. Yeah. Okay, sure. Yeah, the track. Yes. Part of the paper, our collaborator, I mean, I didn't work on track myself, and I don't think it's in the future direction now. We have so many stuff, but our collaborator in the paper showed the transportation using track, which transports the microtubules, actually. The mitochondria, I'm sorry. And he showed that track is also binding to the same regions on lake. That's your question, right? It seems it's the same, the helix 1, helix 2. But we don't know about the N-terminal part, because this N-terminal part is not existing in C. elegans. So we don't know about that. But track, again, seems to need helix 1 and helix 2 for the transportation. But we didn't characterize this by NMR. But it's a good part. I mean, I was talking with Beat about track, but we don't know yet if we will work on that. Sure. Yeah. Yes. It's not high throughput for sure. So, I mean, if this, I hope I understand the question correctly, but we are not talking about library of thousands of compounds to be tested by NMR at the beginning. This will be so much time consuming, and you will need also sample jet and things like that, which we probably don't have. So what I'm talking about is that coming from already like high throughput screening, like fluorescence assay or something like that, or you have algorithm. In this case, I'm showing you here, we have a theoretical algorithm which predicts ligand for the protein already. And that's what we test. So we can start with like compound 2, and then you start to optimize around that. It's not like a high throughput screening in the terms that you start with 12,000 compounds like what sometimes we will do for a full drug discovery. For example, I mean, I can test as many as compounds as possible, right? As long as you give me protein and ligand. The problem is just it's not so efficient time-wise to do NMR on thousands of compounds. If you come to me with 1,000 compounds, I'm ready to do that, I'm fine. But the problem is that this will take really thousands of days as well to investigate. Because, I mean, we don't have a sample jet on the machine now. We will have one in the summer, hopefully. I mean, it's not super advanced one as well. But you can put, I mean, that's what I did in France. We had like 96 rack, and you can put 96 compounds to be screened at the same time. So if you consider this high, it's kind of high. It's okay, 96. But thousands of compounds for NMR, I would say, no, that's not us. Cool. Thank you so much. I appreciate it. Thank you.